Hi, welcome to the tutorial for lesson 2 for functional annotation. So here we'll be working out of the topic functional annotation repository in the BioVCN.net GitHub repository. To launch the tutorial, you'll click this link here to open up Binder, which I have over here, which is now opened. We're going to be working in the terminal environment, which you can access by double-clicking this. And we're going to work through this tutorial together, where we're going to be looking at how BLAST and Diamond functions on an example database or data set. Okay, so the first thing we want to do is ignore that error that comes up. And we're going to make a directory to store our sequences that we're going to be working with called BLAST example. We're going to move those, which are both FAA files, into there, and then we're going to CD into BLAST example ourselves. And if you hit LS, we're working with two FASTA files, both in protein space, so they're annotated as .FAAs. The first one is Cyanobacteria tmed155, which is a metagenome assembled genome from the Mediterranean Sea that has been determined to be a cyanobacteria. In the file is 1,585 predicted proteins. Based off of CheckM, which you can learn more about in the metagenomics topic. It's estimated that this MAG is about 65% complete with a fairly low amount of redundancy / contamination, however you want to look at that value. So it's not that complete, but it's a little bit complete. And we're going to be comparing this genome to the carbon fixation marker gene database. So this is a database that I created myself. It contains 326 proteins chosen to represent the key marker genes of the 5 established carbon fixation pathways, specifically Calvin-Benson, reverse TCA, Wood-Longdahl, 3-hydroxypropionate bicycle, 3-hydroxypropionate, 4-hydroxybutyrate cycle. And so what we're going to do is we're going to walk through this situation, our tutorial. And what we need to do first is we need to create a index for BLAST, which it'll run on our subject sequences. So these are sequences we want to compare against. We're going to take our genome and compare against them. BLAST doesn't care which data set is the which is indexed here, and it might make sense in some capacity sometimes if you're going to compare multiple different databases to this one genome to make the genome the database. But a lot of times it's what you want to compare your sequence to. Right, so in this case, we're going to make a database out of the carbon fixation genes. So we're going to just run make BLAST DB. So we have to explicitly tell it which file we're working with, and we have to tell it which type of file, what type of sequence is in the file. So in this situation, we're working with proteins. If it was nucleotides, we would say nuke for that. And we run that, and that creates an index. And when we hit LS, we can see that that index consists of 3 files that have these .p, PHR, PIN, PSK, which represent our indexes for BLAST. And so we can run our BLAST P command because we're comparing proteins to proteins, which comes installed as part of BLAST when you install BLAST. And so to avoid typos, I will copy and paste what's available in the tutorial into the binder. But let's go through what the various inputs look like here. So for BLAST, we have our query sequence, which is our cyanobacteria, compared against our database, which are carbon fixation markers. We have some out file that we're going to name, which we're going to call BLAST full output. And then we have some things that we can set ourselves. So here, we're setting an E value. So the default number that BLAST wants to set here itself is 10, which is incredibly high and will give you lots and lots of false positives, as BLAST will basically give you any type of small word match that it can find amongst your query and database that you're running against. Here we're going to set a really stringent E value, 10 to the negative 21, so 0.20 zeros and a 1. And so this might look like a very, very small E value for folks who are used to running this against very large databases. But the E value that you get back, so this is the expected value that is returned that gives you some indication of how significant the hit is for your BLAST output, is dependent on the size of the database you're comparing against. So if you're comparing against a really large database, it can be really hard sometimes for BLAST to find a significant match because there's just so much information that it's comparing against in that all versus all search. And so if you're comparing against an R, to consider something for maybe as a potential match, you might have a setting of 10 to the -3 or 10 to the -5. But in a small database like this one, we only have 326 proteins. It's really easy for BLAST to find superfluous matches that don't actually have any biological meaning to us. So we set a really low value for this. Something to take into consideration, and you might want to use going forward, is that bit score, which is a value that BLAST provides, which is based off of the E value that the match that you have gets, as well as the size of the database, takes that database size into consideration. So you can actually use bit scores to compare across databases of different size to see how good of a match you have, quote unquote, how good of a match you have. And so that might be a number that you become more interested in the more you work with BLAST. We can also set the number of descriptions and alignments that are produced here. This is set at 5 just so we don't overwhelm the output. And so like I said, I'm going to copy and paste to avoid typos. Spent so long making sure that this worked. The first time I ran it in Binder and tried to type it out, it did not work. So we're going to hit enter, and this is going to take a little bit to run. And that what we're going to do is we're going to take a look into this BLAST output. So if we just did less and BLAST the full output.txt, what you'll see is a lot of information about how we didn't find any hits. And we can hit tab or spacebar to go down fast through all this. And you can see for a lot of the proteins that are being queried, there are no significant matches in the database that we're searching. That's fine because the database is fairly limited. It's just key marker genes and carbon fixation. If we want to, we can jump to a specific area to look at a match to walk through what that would look like. So here I'm going to jump to the line 11,093. So here we have a match. This is the subject that it's hitting, the ID, as well as the description of the subject of the file that we're looking at, the length of the input that we're, the subject that we're hitting. And here we get values like our bit score, our E value, which again is fairly low, 10 to the -24. But when we start getting into the actual details of this, we'll see that this is actually not that great of a match for this particular set of proteins, this HSP. And so what we're seeing here is the number of identities. So these are the direct matches. There's a one-to-one relationship between this P and that P in the subject, this L and that L in the querying subject. And what it says here is that for identical matches, we have 26% identity, which is very low. There's also this value here called positives. And what BLAST does is it takes into consideration the fact that some amino acids are good substitutions for each other. They have similar size, they have similar polarity. And so that a substitution from one to the other might not have as large of an impact on the protein and the functionality of the protein as a match that isn't related to each other. And these get scored as positives. And so if you include those throughout this alignment, our number goes up a little bit. It's a 42% match at that point if you're allowing some of that flexibility. And so you might hear people compare this between identity, which is an exact match, and then similarity, which takes into consideration those positives, and then the number of gaps that we need to align our query sequence with our subject. And so there's 24 gaps. You can see them here that are occurring predominantly in our subject, but also in our query, but also in our subject. So overall, this is not a very good match. This output is really nice to look at as a person. We have all these nice little summaries of how many numbers versus the total, and summarized in parentheses with a percent value. Computers hate this, right? It's almost impossible to parse. So one of the things that we can do is run BLAST in a computer-friendly manner where we're going to actually output a tab-delimited table. And so to do this, we're going to run basically the same BLAST command, same E value. We're going to change this because of the output format. We no longer have alignments that we're generating and descriptions. Instead, we're just having the total targets that we're returning. So here we're going to set the max target sequences to 10. And then we're going to run this out format version 6. So this is the 6 is the tab delimited type. If we click here, I'm clicking a new tab, we can see that generally, if you use a tab output, output format 6 returns 12 values, but we can adjust that to what we want. So here we're only returning 10, and you can order this in any situation in any order that you wanted to. So here we have a query seek ID, query start, query end, subject seek ID, the length of that subject, which might be important for keeping track of how good of a match you have. The alignment of the subject, start and end, your bit score, your percent identity, and your e-value. So again, I'm going to highlight all of this just to avoid typos, copy and paste. Again, it's going to run. It's going to take a second. And now we can take a look at this output, which we are calling blast output.tab. And so it's, I'm going to make this a little bigger and redo this. So here it's just a simple blast or tab output where our sequences that have matches are ordered in with the parameters that we gave it. So here's our percent identity column here. You can see there's a lot of low percent identities in the 30% and some that are really high. And so what we might want to do is we might want to filter to look at just like the high matches. And so we can do that using some fancy Unix outputs. So here what we're going to do is we're going to use awk, which you can see more about in lesson 4 of the basic Unix tutorial, where we're going to say print the line from column 9 if it's greater than 50. And so here we're using 50%, so 50% identity, which is good, right? It's not incredibly high. This would allow us to see things that are homologs that are fairly distant to each other and allow us to make a decision about if something is accurate or not. But at the same time, it's not all the way at the bottom, that 30% range, which might not have any real biological meaning to us. And so we can take this, we can copy, we can paste. I'm going to make this just a little bigger for us. Maybe we go like this. There we go. And here we have that same table output, but now we're only capturing things that are above 50% complete. You can see that in our table output here. We can sort this, right, by piping our output to sort and saying sort by numerically on column 9. And so here, instead of somewhat random numbers, we can actually order from our most abundant, or our highest percent identity to our lowest percent identity. And so you can see this range of bit scores. So a really high identity value has a really high, close to 1,000, really on the grand scheme of things. And that the lower the identity, the lower that bit score gets. Okay, and so we might be interested in trying to figure out, okay, well, what do these matches actually mean? But right now, all we have access to is our query identifiers and our subject identifiers, our subject IDs. I can tell you, this isn't always the case, but in our particular database that we're using, in the FASTA headers, we have our identifiers, which is what BLAST is using to organize this, but we also have the annotation that goes along with that sequence as well. And so if we wanted to, we could try and match up our identifiers with those descriptions to see a better idea of what these, in theory, really what we're looking at is one protein with multiple successful matches and then another protein with just one match. And so we want to try and figure out if we can look at what those look like. And so all I'm going to do here, again, I'm going to copy to avoid some degree of typos, but we're going to run awk like we just did to find the columns that are high. We're going to use cut to just print out the first column, which is our query identities, or query identifiers, our subject identifiers. And then we're going to sort on our new column 2, which will be sorted on our subject identifiers. And we're going to store that in a temporary file. So we'll just do that. I'll hit enter. We can take a look at that temp file, which now it just has 2 columns in it, our query IDs and our subject IDs. And so now we want to isolate our subject IDs, which we can just do by using cut to print the second column. So now we just have the genes that we're trying to match, their IDs in a separate file. And then we can use grep and its file flag to go through the carbon fixation markers and pull out all of the header lines that have a match to those subject IDs. Then we'll run it through sed to remove the caret, and then we'll sort it based off of that first set before the space. So our identifier, so it'll be sorted by our subject identifiers, which is how we sorted our BLAST matches here. So we'll grab that. This is another temp file. And then we can paste that information together. So that's taking our 2 tab delimited column files, and we're going to paste them together and create one final output file. And then we can take a look at that output file collectively. I'm gonna make it very big because I know how big this is. TMed blast TSV. And so here, what we're looking at is an ordered, somewhat ordered for us output. So it's ordered on our protein numbers that we're looking at. And then the identifier that we're matching to, which we haven't, which is duplicated because of the two tables that we were working with. And then some annotation that was linked to that first, to the subject that we were hitting. So in this instance, protein 60 on this contig hits ribulose bisphosphate carboxylase small chain, or Rubisco small chain, and that there's multiple copy or multiple hits for this 61 protein hits against ribulose bisphosphate carboxylase large subunit. So Rubisco large subunit. So the two necessary components for Rubisco, small and large, are present in this one organism, which would suggest that it potentially has the capacity to fix carbon using the Calvin cycle. So we're going to take a second here. We're going to remove our temp files. And then we're going to move on to how we would do this using Diamond, which is actually going to, you'll see, looks very similar to how we walked through it with BLAST. So the first step is we need to make an index. And so we can do that with Diamond make DB. We're going to have it give it our input, which is our carbon fixation markers. And then we're going to tell it the name of the file that we want to create. And here I'm just going to remove the suffix, because when I run this, you'll see that one of the outputs that it generates, or the output that it generates, the index, it appends a .dmd, DMND suffix to the file name, right? So you can name it whatever you wanted here, but to keep track of what the source is, I just removed that .faa file. And so this is what it will look for, is its database. And so let's walk through Diamond. So in Diamond, you call Diamond the program, which particular type of BLAST you're running. So in this case, BLAST P. You can set the number of computers that are running on this, number of threads, number of CPUs. So here we're just going to run it on one. If you had access to more, you could increase this number to the number of CPUs you wanted to run. Here we are having input of our query sequence, our database sequence, our output, which I emphasize is going to be in fast mode. I'm going to talk about that in a second. The max number of target sequences. So by default, the number for this in Diamond is fairly low. It's about 30. And so if you're running against a big database and you really want to capture all of the potential high quality matches, you might need to increase this. In this instance, we don't need to do that because we're running against a small database, so I'm going to use a smaller number. Changing that number actually also changes the amount of time it takes to run. If you're asking it to return 300 instead of 30 matches, if they meet the quality threshold that you want to use, it will just take longer to find all of those matches and give them back to you. We're going to output it in the format 6. Again, it's going to be tabular, just like we did with BLAST. We're using the exact same order and the exact same setup of values that we'd like it to return. So I'm going to copy and paste this. Oh, before I do that, let's talk really briefly. So I can run Diamond help. So this is the help file for Diamond. Comes out with all the other parameters you can run. A lot of these mirror ones that are similar to BLAST. By default, Diamond runs in the fast mode. There are several other formats that you can run, including sensitive and more sensitive, which increase the level that their Diamond runs to find and detect matches, but it increases your runtime. And so this will demonstrate that by comparing our fast mode with our search with the more sensitive mode for the same search that we're going to run between our cyanobacteria and the carbon fixation database. So again, I'm going to copy. I'm going to paste. I'm going to let it run. We'll see here that the total time it took to run was 0.236 seconds, and it returned 41 high scoring pairs. If we change that setting, so all I've done here, it's the exact same setup, but before this -p, I've added -- more sensitive. So this will be a more sensitive run of Diamond. I'm going to copy and paste that. And you'll notice right away that this runs significantly more slowly. You'll see it iterates multiple times. And compared to that 0.236 seconds, we're now running 1.13 seconds. So about 6 times longer to run that search. the number of HSPs is different, and we're going to cover that in just a second. And so you can imagine how more sensitive might give you, a better match. But at the same time, if you're doing this with a lot of sequences of the really big database, you're also going to be somewhat defeating the purpose of Diamond, which might be to have it run in fast mode to get you your results back quickly. So there's always going to be that trade-off when you're working between speed and accuracy in a lot of these tools. And so approaching it from that perspective is always really important. So let's do a quick comparison of the outputs that we got out of this. So if we did word count -L and we did our original BLAST output in tab mode, there are 40 matches in our BLAST output. If we do word count -L and now we do tmed diamond fast, which is our fast output, and say, well, how many high scoring pairs were there for that particular search? Diamond actually found 1. And then if we go up and we do it and we do more sensitive, and I hit that, we see that that number, that 58, is Diamond found 58 high scoring pairs. And so this goes back, if you watch lesson 2 for functional annotation, we talked about Diamond and how Diamond is similar to BLAST, but it doesn't always return exactly the identical match to BLAST. And so we'll see that that actually can play an impact here on what we're going to see in our output. But that's something to be cognizant of, is that while it's running in a similar capacity to what BLAST does, it's doing local alignment searches, it's giving you an all mapper return value. Those matches, the output doesn't actually match up one-to-one in a lot of instances. And that's just because of the way the algorithm runs under hood compared to the two different versions of the program that we're looking at. So if we wanted to, we could take a look at the types of files that are being output, or not the types of files, the types of matches that we're getting from Diamond. So I'm gonna run this. So I'll skip that word count part for a second. I'm gonna hit enter. Didn't need to go all the way that far. And so you can see here, if you remember from our BLAST output, that we're virtually getting the same results. That's the limit right there. So we were looking at that contag 61 as the protein, and then 60. So again, we're capturing most of that here in our output. You can see here that again, we're looking at just the things that are above 50% complete. You can also see that the bit score values are actually very similar here, right? Which is something that's conserved between Diamond and BLAST, how they're calculated. But you can actually also see that there's another protein that's appeared. It's on a different contag. It's protein number 4 in that contag, and it actually has a different match, or a different hits to a new set of subjects, and has a fairly low percent identity associated with that, and low bit scores as well, respectively. And so we can run this and say, well, how many are here? And we'll see that there are 18 in that particular file. We can also hit up, we can change our fast to more sensitive and get that output. And so if you look here, you'll see that this output actually looks fairly similar to each other. And when you run that word count, it's actually the same. So despite the fact that BLAST in its different, or Diamond BLAST in its different parameters of fast and slow had different outputs, when we actually get to that point where we're including information of where we want our accuracy to be, when we set it some type of cutoff, it actually is returning the same high quality matches that we would expect and want to see as part of this. And so if we keep moving forward with that, we can basically replicate the output that we generated previously, where we want to compare the descriptions. This would allow us to see what that new match might be. And so we can take that information. And so again, this is going to look very similar. These are the exact same commands. I've just changed the files that we're working with. We're going to create one temp file. We're going to grab the subject identifiers and get those into a file. We're going to use grep to grab the descriptions. And then we're going to combine them using paste to get one final output. So I'll copy and paste that. I'm going to make it bigger. And then we're going to look at that output. So here we want to find our diamond matches TSV. We'll look at that. So again, we have the same order as we did in our BLAST output, where we're seeing the same kind of matches. So we again have protein 60 matching the Rubisco small chain. We have protein 61 matching the Rubisco large chain. And then here we have this new protein that was detected as part of diamond, _4, which actually both of these hit a functionality called Formate Dehydrogenase beta subunit. And so we can talk about the significance of that type of discovery, right? Diamond returned a different set of matches with a different level of significance. One of those matches seems to suggest that it's a protein in a different one of these carbon fixation pathways. And this is where functional annotation actually hits the rubber in the road, where we have to start thinking about metabolism. And it's part of the reason why functional annotation is so hard. And this is going to I'm going to iterate this in lesson 3. I'm going to talk about this in all the other databases centric lessons that we're going to have going forward. And that's this idea that a match doesn't always give you the solid answer that you need, but it gives you that first step to go and explore a little bit further. And so let's give an example of this situation, right? So formate dehydrogenase. So this is an enzyme that is not only present in the marker genes necessary for carbon fixation, but it would be also in other pathways that have to do with the cycling of formate in the cell. In this particular instance, we're only hitting the beta subunit. And so formate dehydrogenase has multiple subunits in it. And so to actually have a functioning enzyme, we would have to successfully have formate dehydrogenase's alpha, beta, gamma subunits as part of this particular enzyme. And so the fact that we only hit one very poorly might be an indication that this isn't actually a good functional annotation to reflect back on our particular genome that we're looking for. The other thing is to consider all the context that we have that goes into this. So we said at the beginning that we were fairly confident that this is a cyanobacteria. And we notice, and for those of us who work in the ocean, we notice cyanobacteria, or in lakes, that cyanobacteria are fixing carbon using the Calvin cycle, so we'd expect to see Rubisco. So if I had this MAG, which was incomplete, 64%, and I only hit the small Rubisco subunit, I would use that as an indicator to think, okay, maybe I've successfully found that this particular cyanobacteria has the functionality that we believe of carbon fixation. If I was in the human gut and I had a bacillus MAG that had a poor match, maybe it was in that 50% range, to the small subunit of Rubisco, and it didn't have the large subunit, it didn't have the rest of the Calvin cycle, I would become suspicious that potentially that sequence is in the wrong spot. And so getting a handle on functional annotation and getting a raw result can be fairly straightforward. It involves a BLAST, it involves a database. You can get an answer really quickly, but to frame it in context requires understanding cellular pathways, metabolisms, and what you would expect in particular organisms and what might be unique and what might not be. So that's really the hard part of functional annotation, and it'll be something that we get into. So one of the things that we could do with our current cyanobacteria is, okay, so we have indication that Rubisco is present. We might want to go deeper and see how complete the Calvin cycle is, the CBB cycle is. If we did a search and we only got one match back and it wasn't some of the required elements of the Calvin cycle, we might become suspicious that we don't have enough information to say that this cyanobacteria has the potential to fix carbon. There are a number of tools that allow us to go a little bit further and look at very broadly results like this to get an idea. But at the end of the day, when we get to a match, we always end up having to do extra work to confirm what we're looking at is genuine. And we'll talk about that in future lessons. But thank you for joining me in this particular tutorial, and we'll be back with lesson 3.